In this video, we'll study about immunoglobulins. We'll study history, definition, structure of immunoglobulins, immunoglobulin classes, the structure, physical and biological properties. Let us start with the definition. Immunoglobulins are a group of glycoproteins present in serum and tissue fluids in all mammals. They are produced in response to foreign substances administered into the body with which they react specifically and in an observable manner. So in the definition, what is most important to remember is the term glycoproteins. Glycoses meaning carbohydrates, proteins. So carbohydrates along with the proteins, they are called as glycoproteins. Second important highlighting word is serum. Okay, so the antibodies or the immunoglobulins, they are found in serum. Third important point is they are produced in response to foreign substances. Only they are produced in response to foreign substances and they react with it specifically and in an observable manner. Let us start with the history. For that, we have to separate the particles in order to study that particle in detail. So, how are we going to separate the immunoglobulins? Immunoglobulins, for the first time, they are separated by fractionation method by half saturated ammonium sulfate, in which we get two fractions. They are soluble albumin and insoluble globulin. So this insoluble globulin was further separated by distillation Cabot by using electrophoresis. So what is electrophoresis? Electrophoresis is the method of separation of particles based on their electric charge as well as their molecular size. So the globulins are further divided into three types, are further separated into three types as alpha, beta and gamma globulins. It is observed that the antibody activity is found in the gamma globulin fraction and hence that is called as immunoglobulins. So to see the historical points in detail, how Tisselis and Cabot in 1939 carried out the experiment with rabbit serum. First of all, rabbits, they are inoculated with a ovum protein, and this ovum protein acts as an antigen in a rabbit's body. So rabbit's body produced immunoglobulins, and that is why it is called as immunized rabbit serum. Then they got its separation in the form of two major fractions, as one is albumin fraction and second is globulin fraction. The globulin, as we see in the diagram, is separated into three different parts as alpha fraction, beta fraction and gamma fraction. So the antibody activity is shown in a gamma fraction and therefore they are called as gamma globulins. So to remember this, we must remember the two very important terms as plasma and serum. Plasma is blood minus blood cells, whereas serum is that fraction, that watery portion which comes out after coagulation of the blood, that is after clotting of the blood. Immunoglobulins, gamma globulins and antibodies, the three terms meaning, though they represent the same terms, but in reality their meaning is different. First we'll see about immunoglobulins. The term immunoglobulins, globulin fraction indicates about the chemical nature, whereas immuno represents that the substance which gives resistance to the body. Gamma globulins, the globulin again is a protein fraction. The gamma portion of the globulin is responsible for showing the antibody activity and therefore they are called as gamma globulins. The third word is antibodies. Anti is against and bodies is foreign bodies. The substance which is produced against the foreign bodies and therefore it is called as antibodies. So the antibodies represents a biological nature, whereas immunoglobulin represents the chemical nature. All antibodies are immunoglobulins, but all immunoglobulins may not be antibodies. Right? We'll see the reason behind that, why it is called so at the end of the video. To start with the structure of immunoglobulin. If you look at the diagram carefully, you'll observe that immunoglobulins, they are made up of four chains. Two are short chains and two are longer chains. These short chains, they are called as light chains, whereas the larger chains, they are heavy chains. This is the Y-shaped model of immunoglobulin. So if you see at the top portion of the structure, we will find that there are amino groups. Therefore, this terminal is called as amino terminal. Whereas at the end of the terminal, we'll find that there is COO, that is carboxyl terminal, and therefore it is called as carboxyl terminal. It has carboxyl group, acidic group. So the antibody activity or the antigen binding ability is found in amino terminal, whereas the actual biological activity resides in the heavy portion. So this 4-chain structure is a very unique structure, and we have to always remember the 4-chain structure. We observe that, for example, if you observe it very carefully, we see that there are 2 points at the tip of the immunoglobulin molecule referred as FAB. What is that FAB? F stands for fragment, AB stands for antigen binding. So the fragment antigen binding is called as FAB. Second point is FC. FC is fragment, C is constant. Whereas FD is the inner portion of the heavy chain, is called as FD portion. FD stands for variation. Okay. So if you look at the diagram again, we'll observe that there is a red line. The portion above the red line is called as the variable portion, whereas the portion below the red line is called as the constant portion. Variable portion or variable region, constant portion or constant region. Why it is called as variable? Because variable region is the region which shows variation in number and sequence of amino acids, whereas constant region is showing the constant number and sequence of amino acids. So C stands for constant, that is 1, the reference of the C. Secondly, C stands for the fragment which undergoes crystallization. Third is the fragment which is showing complement binding ability. So meaning of the C has got 3 meaning. One is C refers constant, C refers crystallization, and C refers complement binding. So every immunoglobulin molecule will have 2 FAB sites. FAB means the fragment antigen binding. There are 2 FAB sites. There is one FC portion and there is one FD portion. Okay. So where the half of the portion is the variable region as shown in the diagram above the red line, whereas the portion below the red line is a constant region. So if you want to study the molecule in detail, we must know what is the action of different enzymes on immunoglobulins. Okay, we'll see the action of different enzymes such as papain, pepsin, and mercaptoethanol reduction. The first we'll see papain digestion. As shown in the diagram, we'll observe that this particular portion, papain digestion, gives 3 important fragments. There are 2 FAB and 1 FC. 2 FAB portions and 1 FC, that is 2 FAB fragments and 1 FC portion. So the product of papain digestion on immunoglobulin gives rise to 3 portions, 2 FAB and 1 FC. Whereas if you look at the pepsin digestion, in the pepsin digestion also we get the 3 fragments, but there is a little variation, a little addition of the disulfide bonds in FAB portion. So as to differentiate it from the fragments which are formed in a papain digestion, they are called as FAB dash portions. Right? So how many there portions will be? There are 2 FAB portions and 1 FC fragment. Whereas mercaptoethanol reduction shows that 2 heavy chains and 2 light chains as a 4 separate chains they are formed by mercaptoethanol. So once again we'll see what is the effect of different enzymes on immunoglobulins. So papain digestion gives rise to 3 fragments in total. There are 2 FAB sites and 1 FC portion. Whereas pepsin digestion gives 2 FAB dash and 1 FC fragment. And mercaptoethanol reduction gives rise to 4 chains as 2 heavy chains and 2 light chains. We'll see in detail about the light chains. Why they are called as light chains? Because they are smaller chains, the molecular weight ranging from 20,000 to 25,000. Amino acids are ranging from 210 to 230. They attach to heavy chain by disulfide bond. SS bond is called as disulfide bond. As we have seen in the diagram, there are two small chains and two large chains. So the two small chains, they are called as light chains. There are two types of light chains as kappa chains and lambda chains. And these kappa and lambda chains, they are found in 2:1 proportion. Why they are called as kappa type and lambda type? Because they are named after the scientists Corngold and Lapari, and therefore they are called as kappa type and lambda type. These immunoglobulins may have either a kappa type or lambda type of light chains but never both. Meaning is what? In a given immunoglobulin, there are two light chains which are of kappa type or there are two light chains which are of lambda type, but it will never happen that one is kappa type and one is lambda type. So we have to remember this, that immunoglobulin has got either kappa or lambda type of chains. Coming to the heavy chains. They are larger chains. Molecular weight is about 50,000 daltons. Amino acids ranging from 420 to 460. They join together by again disulfide bonds. Here internally they are joined with light chains with a disulfide bond, but internally two heavy chains, within the heavy chains there are again disulfide bonds. Heavy chains are structurally and antigenically different. Depending on the antigenic property of the heavy chains they are divided into 5 different types. And based on the heavy chain different types, we have different immunoglobulin classes. For example, as shown in the table, IgG represent that these heavy chains, the antigenic type on the heavy chain, they are represented by the Greek letters. For example, IgG. G stands for gamma type of heavy chain. Therefore, it is called as IgG. IgM, M, because why M? Because the antigen determinant on the heavy chain is of mu type and therefore it is called as IgM. IgA, the antigen determinant on the heavy chain of this type is of alpha and therefore it is called as IgA. IgD, it is because the antigen determinant on the heavy chain is delta and therefore it is called as IgD. IgE, because the antigen determinant on the heavy chain is epsilon and therefore it is called as IgE. So once again we'll repeat, the antigen determinant on the heavy chain is responsible for classifying the amino acids into the different classes such as IgG, IgM, IgA, IgD and IgE. This is on the basis of the antigenic type on the heavy chain and which are referred as per the Greek letters as gamma, mu, alpha, delta and epsilon. Okay. We'll see the hinge region. What is that hinge region is? Immunoglobulin, the 4-chain structure. Why it is called as 4-chain structure? There are two light chains and two heavy chains. And if you observe that the 4-chain structure, if it is folded, and this completely folded chain structure is expanding, or it is that particular region which is responsible for swinging out the arms, that particular portion is called as the hinge region. And that hinge region is responsible for the taking the different types of shapes in a immunoglobulin molecule. Hinge region has large number of proline residues. And this is very important point to note, that the hinge region is very rich in proline amino acids. So once again, before we go into the details of the immunoglobulin classes, we'll see once again revise the entire immunoglobulin structure. For example, the immunoglobulin is having two light chains, two heavy chains. That is, it is a usual 4-chain structure. In every immunoglobulin, there are two FAB sites. This is one FAB portion, is another FAB portion. That is, one immunoglobulin has got two FAB sites. Then there is the FC portion at the bottom. Why it is called as FAB? FAB, the fragment antigen binding. Whereas FC stands for the fragment undergoing crystallization. The fragment showing the constant number and sequence of amino acid, and the fragment which is fixing to the complement. So in every immunoglobulin we observe there are two FAB sites and one FC portion, whereas the inner side of the heavy portion, which is joining to the light chain, is called as the FD piece. FD stands for variation, the fragment which shows variation in number and sequence of amino acids is called as FD portion. The portion which is the upper portion, the upper surface or upper region is called as the variable region, whereas the lower region is called as a constant region. So based on why it is called as variable and constant, variable is with respect to number and sequence of amino acids, whereas constant is the constancy with respect to the number and sequence of amino acids. As we see in the hinge region, we will find that these arms of the immunoglobulin molecule, they are swinged out, and the swinging out is possible because of the bending nature of this portion region, just like it works like a hinge of the doors, right? And therefore it is called as the hinge region. This hinge region have large number of proline residues. So let us see what are these immunoglobulin classes. And as we have seen that based on the heavy chain antigenic determinants, the immunoglobulins are classified into 5 different classes such as IgG, IgM, IgA, IgD and IgE. Ig stands for immunoglobulin. Immuno, I is for immuno, G is for globulin and G is for because it is having the gamma as the antigen determinant of the heavy chain, therefore IgG. Okay, so let us see in detail about these classes, their properties in detail. So we'll see the details about the IgG. General properties we'll see, physical properties, chemical properties and biological portion. As seen in the diagram, the IgG is the usual 4-chain structure, right? So this molecule is having the observed or is found in a monomeric form. Monomer is there are 2 light chains and 2 heavy chains. That is called as a monomer. Usual 4-chain structure of the immunoglobulin is called as a monomer, right? Most important point to remember about IgG is it is a major immunoglobulin in normal serum. About 70 to 80% of the total immunoglobulins, IgG constitutes about 70 to 80%. Okay, and that is why it is called as a major immunoglobulin in normal serum. Molecular weight is 1,50,000 Daltons. Sedimentation coefficient is 7s. The S stands for Swedberg's coefficient. Half-life of this immunoglobulin is 23 days. And that is why 23 days is a long time. The long-lasting antibody. Okay. Another important point to the property is it contains less carbohydrate. Very less. About 3% of the carbohydrate is found in IgG. Valency. Most important part of the immunoglobulin is valency. What is that valency is? Valency means how many antigens will be bound to one immunoglobulin, that is called as a valency. So if you look at the diagram, each immunoglobulin can bind with 2 antigen molecules because there are 2 FAB sites. Monomer, that is a single immunoglobulin, has got 2 FAB sites and therefore the valency of IgG is 2, right? So once again I will say that one immunoglobulin has got 2 FAB sites. In the sense FAB is fragment antigen binding. There are 2 FAB sites and therefore the valency of immunoglobulin is 2. This IgG is 2. Normal serum level of IgG is found to be 8 to 16 milligram per ml. There are 4 subclasses of IgG as IgG1, IgG2, IgG3 and IgG4. Most important point to remember about IgG is it is a major immunoglobulin molecule found in the serum in the amount of 70 to 80%. Second very important point to note is half-life time of IgG is 23 days, right? And the amount in the normal serum is 8 to 16 milligrams per ml. Okay. Moving to the other properties of the IgG, it is a major immunoglobulin synthesized during the primary immune response. It is distributed intravascularly and extravascularly. What do you mean by intravascular and extravascular? Within the vessels and outside the vessels, Within is intravascular and extra is outside the vessels. It has got greater antigen affinity than IgM. These are the physical properties. Coming to the most important part is biological properties. Very, very important property of IgG to note is it crosses placental barrier and provides natural passive immunity. So what do you mean by this? When the baby is in the womb of the mother, then the baby gets the entire nourishment from the mother to the fetus. Okay, so this placental transfer or mother to the fetus transfer also transfers IgG, and that is how the fetus gets the resistance to fight against the infection. This particular type of immunity, this particular type of resistance the fetus gets, that is called as a natural passive immunity. And this is the only immunoglobulin. IgG is the only immunoglobulin which can cross the placental barrier and provides the fetus natural passive immunity. So it is a very important property to note. Second important is enhances phagocytosis. What is phagocytosis? Phagocytosis is engulfment of a solid particle is called as phagocytosis. Here, engulfment of solid particles meaning is what? Engulfment of the antigen, the foreign material, that is phagocytosis. It can activate complement and attracts PMN by chemotaxis. Right? PMN is what? PMN is polymorph nuclear cells, that is neutrophils. It participates in complement fixation, precipitation, and neutralization of toxins and viruses. Overall, if you look at the function of the IgG, we'll find that IgG protects the body fluids. So once again, we'll see the important highlighting features of IgG. Number one, IgG is the major immunoglobulin found in our serum, about 70 to 80%. Number two is it has got molecular weight of 1,50,000 Daltons. Number three, it has half-life time is 23 days. Number four, very important to remember, is normal serum level is 8 to 16 milligrams per ml. Another property is, coming to the biological properties, the very, very important to note and remember is it crosses placental barrier and provides natural passive immunity. It enhances phagocytosis and activates the complement and attract polymorph nuclear cells. And overall function of IgG is protects the body fluids. So if someone asks you about the IgG, then the function, overall function of the IgG is it protects the body fluids. Sometimes the question is asked in examination about define immunoglobulins and describe the structure of immunoglobulin and highlight the properties of IgG. The 3 points we have to write is the definition we have to write, then coming to the diagrammatic presentation, followed by the description of the structure of immunoglobulin, and third part is IgG, highlighting the feature, the physical properties and the biological properties, and ending with the overall function of the IgG, that is it protects the body fluids. We'll see the second type of major immunoglobulin, that is IgA. A stands for what? A stands for the antigenic nature of the heavy chain of this immunoglobulin is found to be alpha type and therefore it is called as IgA. This, as is shown in the diagram, we'll find that this immunoglobulin is existing in the form of either a monomer or a dimer. Right? Monomer is a usual 4-chain structure, or a dimer, if you look at, there are 8 structures where additionally there is the 1 point. We'll see this in detail. Okay. Coming to the general properties of IgA, it is the second most abundant immunoglobulin in normal serum, ranging from 10 to 13% of the total serum. It is a monomer or dimer. Molecular weight is 1,60,000 Daltons. Sedimentation coefficient is 7s. Half-life time is 6 to 8 days. Contains less carbohydrate, but comparatively as compared to the IgG, it is having more carbohydrate, 11%. Valency is 2 or 4. Now why it is called as 2 or 4? If you look at the structure, you will observe that in the monomer, there are 2 FAB sites. Therefore, valency is 2. Whereas in a dimer, there are total 4 FAB sites, that is FABs, antigen binding sites, and therefore the valency is 4. Normal serum level is 0.6 to 4.2 milligrams per ml. There are 2 classes of IgA, IgA1 and IgA2. So let us try to remember these properties in comparison with the IgA. In IgG, what we have said, the first point is it's a major immunoglobulin consisting of 70 to 80%. Here in IgA, it is 10 to 13%. IgG exists as a monomer, but IgA exists as a monomer or dimer. Molecular weight is 1,50,000. In IgA, it is 1,60,000. Sedimentation coefficient is same in both. Half-life time, very important to note, is IgG has got 23 days, whereas IgA has 6 to 8 days. IgG has only 3% of the carbohydrate, whereas IgA has got 11% carbohydrate. In IgG, there are only valency is only 2, that is, it can bind only 2 antigens, whereas in IgA, it can bind to either 2, if it is in monomer, or it is a dimer, it can bind to 4 antigens. Normal serum level is 8 to 16 milligrams in IgG, whereas in IgA, it is 0.6 to 4.2 milligrams per ml. IgG has got 4 subclasses as IgG1, G2, G3 and G4, whereas IgA has got 2 subclasses as IgA1 and IgA2. Moving to the very important point to note is that IgA exists in 2 forms. Just now we have said that it is either a monomer or a dimer. Monomer is a serum IgA, whereas the dimer is a secretory IgA. Right? This secretory, why it is called a secretory IgA? Because this immunoglobulin is present in secretions such as saliva, tears, nasal fluids, sweat, colostrum, secretion of lungs, genitourinary and gastrointestinal tract. Because it is found in secretions, this immunoglobulin is called as secretory IgA. Second, as I have said, it is a dimer containing 4 heavy chains and 4 light chains. Molecular weight is, obviously it will be more because it is a dimer, 3,85,000 Daltons. Sedimentation coefficient is 11s. It possesses a cysteine-rich polypeptide chain called as J chain. J stands for joining, right? And because of these, 2 molecules will be joined together with help of J chain. It also possesses glycine-rich polypeptide called as a T secretory or S secretory piece. It helps in transport across the cells. Coming to the biological properties of IgA, it is present in secretions. It provides local immunity against the respiratory, intestinal and urinogenital pathogens. So if you look at the biological properties, we'll find that every immunoglobulin has got its own unique function. As we have said that in IgG, it is protecting the body fluids, whereas IgA protects the secretions. So it is providing local immunity to respiratory tract, to the intestinal tract, to the urogenital tract. Okay. It activates alternate complement pathway. It promotes phagocytosis and intracellular killing. Whereas the overall function of IgA is IgA protects the body surfaces. Right? So if you the question is asked examination about the highlight the properties of IgG and IgA. So first thing we are going to do is we are going to show the diagrammatic presentation of IgG as a monomer, whereas in IgA it can be a monomer, it can be dimer, and then highlight the physical properties, give the importance of biological properties, and at the end the entire overall property of IgG, overall role of the IgA, that it protects the body surfaces. Okay, hope you understood this. Moving to the next very important immunoglobulin is IgM. Why it is called IgM? IgM is M is for mu, because mu is the antigen determinant on the heavy chain of this immunoglobulin, and therefore it is called as IgM, immunoglobulin M. Okay. If you look at the diagram, we'll find that this immunoglobulin has got 5 monomeric structures. So it is a larger molecule, right? And most important property to remember, it is the first immunoglobulin formed in every antibody response. This is the first immunoglobulin which is formed in the fetus at the age of 20 weeks of the fetus. And that is why IgM is participating in a primary immune response. Its normal serum immunoglobulin is 5 to 8%. As we have seen here that there are 10 heavy chains and 10 light chains, and there are 5 subunits of the monomer. Because the immunoglobulin is made up of 5 different subunits, and therefore obviously one monomer is having 2 FAB sites. So 5 will be having 5 into 2 is 10 heavy chains and 10 light chains. Okay. as obviously it bears the CH domain. CHH stands for heavy. Heavy chains, they are larger in number. Right? Domains, that portion is more in number. Molecular weight is 9,70,000 millionaire, and therefore it is called as a millionaire molecule. Because the molecular weight is 10 per millions, and therefore it is a millionaire molecule. Sedimentation coefficient is 19s. Until now we have said about IgG and IgA, they are 7s particles, whereas IgM is a 19s particle. Okay. Another properties of IgM, it is a half-life time of 5 days. Carbohydrate content is 10%. Valency is 10. Very important property to remember about IgM is its valency is 10. Meaning is at a time one immunoglobulin molecule can bind to 10 antigen particles, and therefore it is called as valency is 10. Normal serum level is 0.5 to 2 milligrams per ml. IgM has got two subclasses as IgM1 and IgM2. It is intravascular, purely intravascular in distribution. As in case of IgG, we have seen it is extra and intravascular. In case of IgA, we have said that it is only in secretions, mainly in secretions, whereas IgM is intravascular in distribution, right? That is why it is important to know about biological activity, that its highest valence is 10, it is very efficient in agglutination and cytolytic activity. It is largely confined to bloodstream, and therefore it protects against the bacteremia. What do you mean by bacteremia? Mia is blood, the presence of bacteria in a blood. So IgM is responsible for taking or looking after the bloodstream, the bacteria or any foreign material found in the bloodstream, and therefore very important role of IgM is protection of the bloodstream. It is not transported through the placenta. It fixes complement by classical pathway. It is most efficient antibody in agglutination and complement fixation and cytolytic reactions, as well as it neutralizes viruses and toxins, and therefore it is very important in bringing about variety of different antigen antibody reactions. It is short-lived. It disappears rapidly, and therefore presence of this immunoglobulin always indicates a recent infection. Whenever anyone suffers from a infection, that time if you look at the IgM count, if IgM count is increased, then we can come to know that the person is suffering from the recent infection. Overall function of IgM is protects the bloodstream. So IgG is protecting the body fluids, IgA is protecting the secretions, IgM is protecting the bloodstream, right? Next line immunoglobulin is IgD. It is called as D. D stands for delta. Antigen determinant on the heavy chain of this immunoglobulin is delta, and therefore it is called as IgD. It is found less than 1% of the total. It is very, very less in quantity. It is a monomer having two H and two L chains, two heavy chains and two light chains, or 4-chain structure. Molecular weight is 1,84,000 Daltons. Fermentation coefficient is 7s. Half-life time is 2.8 days, but it contains maximum amount of carbohydrate is 13%. So sometimes the question can be asked, which immunoglobulin shows the maximum carbohydrate? That is IgD. And which is having the less carbohydrate content is IgG, is having only 3% of carbohydrates. Valency is 2. The normal serum level is 0 to 0.04 milligrams per ml. If you look at the biological activity, you will find that IgD along with IgM functions as antigen receptor on the surface of B lymphocytes for recognition of antigens. So main role is in combination with the IgM. It always works in combination with the IgM to identify or to recognize the antigen and the receptors. Second function is it controls lymphocyte activation and proliferation to produce antibodies or suppression. Suppression is to stop the antibody production, that is suppression. Coming to the last type of immunoglobulin, that is IgE. E stands for epsilon. The antigen determinant on the heavy chain of this immunoglobulin is epsilon, and therefore it is called as IgE. It occurs very low in concentration. Before we go into the details, we'll see that IgE for the first time found out by two scientists, Ishizaka and Ishizaka. Right? When they were actually performing an experiment of transferring the immunity from one person to the another person against one of the, you can say, fish. So if they from one person they allowed to take the fish and the antibodies are taken out and the antibodies are transferred to the another person. It was observed that the same type of allergic reaction was also transferred to the second person, and therefore this then the scientists were found out that what was that immunoglobulin is, and that immunoglobulin was found to the IgE. It occurs in very low concentration. It is a monomer having two heavy chains and two light chains. Molecular weight is 1,88,000. Fermentation coefficient is 8s. Half-life only 2.3 days. It contains 12% carbohydrates. Valency is 2. Normal serum level is less than 0.00003 milligrams per ml. It is extravascular in distribution and is inactivated at 56 degrees centigrade. Now the question is why we have said that it is the normal serum level is 0.00003 milligrams per ml, because when you use the word as normal, that means the person is not suffering from any infection or any disease. That is called as a normal person. But if the person is showing some allergic reaction, then the person starts showing production of immunoglobulin IgE, and that time only the immunoglobulins will be observed, will be found in the person's body, and therefore we say that normally the person's serum do not show presence of IgE. If you look at the biological activity, it has affinity towards the surface tissue cells, particularly the mast cells and basophils, and responsible for degranulation of these cells, releasing the vasoactive amines and other mediators, and responsible for type 1 hypersensitivity. What is that? If you have any allergic reaction, the first thing that we observed is there is a itching, there is a redness, there is a oozing out of some material, for example. So these all reactions that take place because of the release of the vasoactive amines. And this release is from the mast cells and basophils. And this release is because of the IgE bound on the surface of these mast cells and the basophils. Second function is it triggers release of the vasoactive amines and chemotactic factors. Right? If you look at the overall function of the IgE, it mediates reagenic hypersensitivity. So that therefore this IgE is also called as reagenic antibody. Right? And this main role it participates in allergic reactions. So if you look at the entire portion as coming to the summarizing the entire points as the historical points, the definition, the structure, and the immunoglobulin classes. The questions that can be asked on this is number one, the structure of the immunoglobulin can be asked in detail for 10 marks. The structure along with any one immunoglobulin for their properties can be asked for, or there can be short notes on IgG, IgM, IgA, Ig, right, D and E together, right? So I hope that you have followed these points, and if you have any questions, you can ask at any time. Okay. It plays important role in immunity against the helminthic parasites. So thank you so much.